Thanks for that introduction, Laura. And thank you everyone for joining me today for this webinar on the Genomics Workbench, Introduction to Genomics Workbench. So before we begin, just a legal disclaimer quickly. The QIAGEN products showcased here are intended for molecular biology applications only, and these are not intended for diagnosis, prevention, or treatment of a disease. And for up-to-date licensing information on your product-specific disclaimers, please see your QIAGEN handbook if you're using other QIAGEN products. So quickly, before I get into the demo, I just wanted to focus on the typical end-to-end NGS workflow. So you have sample isolation, target enrichment, library, some sort of library construction, then you have this sequencing run, and then you have the data analysis, and then you have data interpretation or insight. So these are the typical steps in an NGS analysis. And at QIAGEN, we have tools for all these steps. So we truly have a sample to insight workflow. So we have kits for sample isolation, target enrichment, library. Then we have the gene reader as a sequencing machine, and then we have data analysis tools and interpretation tools. So we have a true sample to insight workflow. And the focus of today's topic, the Genomics Workbench, sits under the data analysis bucket. So, but we do truly have tools for sample to insight, right from sample isolation all the way to interpretation. And then even just for the informatics part, even if you focus on the QIAGEN bioinformatics part, we have tools that will take you from your FASTQ sequence to insight. So even if you start with your raw sequencing reads, we have tools that will take you from that raw reads to biological insight with the three legacy brands as the CLC Bio tools, BioVis, and Ingenuity pathway analysis. And just to focus on the data analysis bucket of our bioinformatics tool suite, we have the standalone applications such as the Genomics Workbench, which is the focus of today's talk. We have the main workbench and the biomedical workbench. And then if you're running high throughput sequencing with hundreds and thousands of samples, we have enterprise solutions such as the CLC Genomics Server. And then, but focus of today's topic is the CLC Genomics Workbench. So the introduction to CLC Genomics Workbench, which is a cross-platform desktop application with a graphical user interface. And my aim for today's demo using the CLC Genomics Workbench is that I want to identify variants that are unique to line A of my cucumber crop as compared to line B. So I have two lines from my cucumber crop, and I want to find out variants in line A which are unique as compared to line B. That is my aim for today. And then after comparison, my other aim is to visualize my uniquely detected variants in my line A in dynamically linked views like the one that you're seeing right now. So I want my variants on the screen as a table, and then I want to visualize those variants in a genomic context with my sequencing reads and my annotations. So that is my aim for today. And while to achieve that aim, this is kind of the agenda that we're going to go through to achieve that aim that I wanted today. So we're going to focus on data import, like how to import my raw sequencing reads and reference data into the workbench. Then we're going to focus on what tools are available to do my analysis and what tools are available overall in the Genomics Workbench, and then how to run those tools. Then we're going to focus on constructing end-to-end workflows. That is a very powerful feature of the Workbench, which allows you to construct end-to-end analysis workflows. And then we're going to see data visualization, like how to create dynamically linked views, as you just saw, so that you have one single view, one single genomic view of all your data, which makes analysis much easier. So before I jump into the demo, what are the features of the workbench, what all you can achieve? So you have reference assembly of genomic and RNA-seq data. You have multiple variant calling tools to generate, annotate, and compare high-confidence variant calls. You have differential expression and statistical analysis tools for RNA-seq and small RNA data. And you have de novo assembly of genomes. You have tools for analysis of epigenetic data. And then you have the QC and pre-processing of NGS tools. That is the usual adapter trimming, quality trimming. Those are QC and pre-processing tools. Workflows, as I mentioned, you can construct end-to-end analysis workflows. Interactive visualization, so the dynamic linked views that I talked about and that we'll see in the demo. And then you have certain external modules available to extend features of your genomics workbench that we call as plugins that you can apply to the workbench. And then quickly to focus on workflow automation, because I want to focus on workflow automation a lot because this is a very powerful feature of the workbench. With with building workflows in the genomics workbench, you can build a sequence analysis pipeline rapidly with no or very limited scripting skills. And then you can standardize and distribute these workflows within your team or outside of your team if you want to. As you can run these workflows on a desktop, or if you have the high throughput solution and you have the genomic server, you can run the workflows on the server also. And you have a lot of flexibility in configuration of these workflows, as you will see in the demo. So at this point, I'm going to jump into the workbench, and we're going to begin the demo. So just before, this is the user interface of the genomics workbench, and it is kind of sequestered. It is divided into three parts. The navigation area that I'm pointing to right now is where your data is going to be. So all the data that you import into the workbench, or all the data that you analyze and you produce results, those result files are going to be in this navigation area. The one on the right is the visualization area. So the one that I'm pointing to right now is where you're going to visualize your data. And then the toolbox here is where you will have all your tools. And these are all the tools in the workbench, and as you can see, they are grouped by features. So you have your NGS core tools, then you have resequencing analysis tools, so all the variant detectors and variant calling tools are under resequencing analysis. Then all the RNA-seq tools and statistical analysis tools are under the transcriptomics analysis toolbox. So as you can see, the toolbox is sequestered by feature. So it's easier to spot the tools that you want to. Now that we have seen the general layout of the workbench, as in step one in beginning your NGS data analysis in the workbench is always you have to import your data into the workbench. So your FASTQ files and your reference sequence. So FASTQ files are your raw FASTQ files, and then the reference sequence and the reference annotations. As in, if you're working with a model organism like corn or soy or mouse, for example, then you have this download reference genome data tool, which you can use to download your reference sequence and reference sequence annotations. But if you're not working with a model organism, for instance, for today's demo purpose, I'm working with Cucumber, which is not a model organism, and which is not available in the download genomes tool. If you have that, if you're working with an organism which is not available here, then you will have to import the reference data into the workbench also. So step one, I'm going to import my reference sequence from a standard FASTA file. And to do that, I'm just going to click on the import tool here. I'm going to go to import tracks. I'm going to type of file to import FASTA because my reference sequence is in a FASTA file. I'm going to select FASTA, and there are other formats available also to import your reference sequence, such as GenBank, for example. But right now, for this demo purpose, I'm going to select FASTA. So types of files to import is FASTA. I select my FASTA file of my reference sequence, and I'm going to save it. And if I now open, it's going to show me that it imported my reference sequence here. So then I want to import my annotations. So now I have my reference sequence. Now I want to import annotations on my reference sequence. And to do that, the steps are the same. I go to import and tracks. And now to import annotation tracks, any standard GFF, GVF, GTF files are supported, which is the usual file format for your annotations or gene models, for example, and that is supported. So I have a GFF file of my annotations, and I'm going to select that. And to import a GFF or annotation file, you need a reference sequence already imported into the workbench so that you can tell the workbench while importing your annotations that this is the reference sequence that I want to import my annotations against. So I'm going to select my reference sequence, and I'm going to save it. And now if I open my reference directory, you can see that my CDS, gene, tRNA, RNA, these are the annotations that I had in my GFF file, have been now imported into the workbench. So I have my reference sequence, I have my reference annotations. Next step is to import my sequencing reads. So to import my NGS sequencing reads, again the same import functionality, and I'm working with Illumina reads for this demo purpose. So I select import Illumina, and I'm working with paired read files. So a couple of things that I wanted to point out while importing. So most times you are dealing with paired read files, so just to make sure, you have to keep this option checked of paired reads to tell the workbench that these are paired reads. And when you select paired reads, the workbench is going to expect two files, that is your forward reads and reverse reads. And I selected two files, which are my forward and reverse reads for that sample. I have my paired reads option selected. This is the paired read information. This is the insert size or the distance between your forward and reverse read, for example. This is your insert size, the minimum distance and the maximum distance that you expect between your paired reads. And then the other options I'm going to keep as default for now, and I'm going to save it in my input directory. I'm going to finish. It's going to, the pop-up is going to tell me that it's done, and these are the reads that it imported right now. And as you can see, the paired in the parentheses after indicates that these are paired reads, and I can even open and actually look at the reads in the workbench. If you want to open the read file in the visualization area, you can, it gives a very nice visualization of the reads. Even with the Q score, you can see the Q score at each base also. So now I have imported my reads, I have imported my reference sequence. The next step is, as in, usually you will run through all the pre-processing steps like adapter trimming or quality trimming, and these tools are available in the NGS core tools. Here you can see you have trim sequences or demultiplexing reads. So all those sorts of pre-processing tools are available. For this demo purpose, I'm not going to focus on that. I'm just going to assume that I have my pre-processed reads. I have done processing on the reads, and my reads are now ready with the adapter trimming and quality trimming. So the next step would be to then map my reads to a reference sequence. So to do that, any tool is going to follow the same procedure that I'm going to do right now. Right now I'm going to run an individual tool. So I'm going to map reads to reference under NGS core tools. I'm going to double click on map reads to reference. And every tool is going to follow the same steps. You launch the tool, and you're going to run through a series of steps in a wizard, and then finally you're going to run the tool. So, for example, in the step 1, in step 2, it asks me to select the sequencing reads that I want to map to my reference sequence. For this demo purpose, I have the reads from my samples, the two samples, my cucumber line A and cucumber line B. So sample 1 is from cucumber line A, sample 2 is from cucumber line B. And right now I do map reads to reference for my line A. I select my sequencing reads for my line A. Go to step 2, it tells me to select my reference sequence. So I select the reference sequence that we imported in step 1. Now it's going to tell me to adjust a bunch of parameters if I want to. And the thing that I want to focus on here is the help that you have for these particular options. So as you can see, if you want like a one-liner information about each of these parameters that are here, there are tooltips that you can hover over the particular parameter. For example, I want to know what an insertion cost is. I hover over. There are helpful tooltips that will give you some information about what these parameters are. But if you want more detailed information about these parameters or about the tool in general, anytime while running the tool, you have this help question mark here, this button that you can click to get the help on the particular tool that you're running. So for example, I want more detailed information about these parameters right now. So I'm going to click on the help button, and it's going to open this help menu for me that gives me help about the map reads to reference tool. And then I want information about the mapping parameters. So I'm going to click on mapping parameters, and then I'm going to get much more detailed information about what these parameters are. So for the demo purpose, I'm just going to keep all the parameters default, but this help option and the tooltip option are two very nice features that give you information about the tool that you're running. So for the demo purpose, I'm going to keep the default parameters, and same thing here. Even output options, if you want to know more, you can always click on the help menu. Right now I'm going to keep it as default, and example, I'm going to save the output to my output directory, and it's going to map reads to my reference sequence. And what I'm going to get is this reads track. So just a quick word on tracks, as in data in the workbench is arranged mostly in the form of tracks. So if you work with the UCSC genome browser, for example, then it's a very similar concept where you have different types of tracks. For example, read map. When you map reads to reference, you produce a reads track. The reference sequence that we imported produced a reference sequence track, and then the annotations that we imported produced the annotations track. And that now I have mapped my reads to a reference sequence. Now step 2 is to call variants. So I have my map reads. Now step 2 is call variants. So same thing, toolbox. Under resequencing analysis, you have multiple variant detectors, and you choose which variant detector you want to run. Like this time I choose a low frequency variant detector. And as you can see, the tool is again going to follow a same pattern as the first one, where you have a wizard and you have to run through the steps. And then you always have the help menu to get information on the tool. I'm not going to run this individual tool right now because, as you know, as I said before, most of the times NGS analysis the NGS data analysis has a series of steps. For example, you have all the pre-processing steps, you have read mapping, you have variant detection. So it is a workflow. And what I want to focus on now is building workflows. So for example, to analyze my data, my both cucumber lines, as you can see, if I were to run individual tools, I would have to run one tool after the other manually for each sample or for each line. But I do not want to do that, because right now I have 2 lines, but tomorrow I can get 50 lines. And then what I want to do is I want to build a workflow and then have that workflow run automatically for each and every sample that I have. So to build an end-to-end analysis workflow, you go to workflows and then you click on new workflow. And then a workflow window is going to open in the visualization area, and you can build your workflow there. And to build for building a workflow, it's very simple. It's a drag and drop functionality from your toolbox into the workflow. So for example, just for demoing how to build a workflow, say my first step in my workflow is map reads to reference. Say, as in I assume every time I'm going to get cleaned preprocessed reads, and every time the first step in my workflow is going to map reads to reference. So go to my NGS core tools, I drag my map reads to reference toolbox tool, and it gets added to my workflow. And then the tools in the workflow window, you can think of them as 3 different layers. So the top layer is the input to this tool. So the input to this map reads to reference tool are sequencing reads, reference sequences, and an optional masking track. Then you have the algorithm itself, and then you have the output. So input, algorithm, output is how you can look at the tool here. So where are the reads coming from? I'm going to indicate that the reads are going to be the input to this workflow. So when I'm going to run this workflow, I'm going to input my sequencing reads into this workflow. And then I run the algorithm, and the output is the reads track. But now I have to build a workflow. So my next step, for example, might be variant detection. So I'm going to add a variant caller as step 2 in my workflow. Again, drag and drop functionality. Same concept, input, algorithm, output. So input to my variant detection is my reads mapping or reads mapping track. I have the algorithm, and my variant track, variant table or report is the output. So where is the read mapping going to come from? It's going to come from my map reads to reference. So the output of this goes to my input of my variant caller, and what I want as the output from this workflow is my variance track. So I'm going to right click, and I'm going to tell the workbench that I want to output a variant track from this workflow. So again, this is just conceptual, just to show you how to build an end-to-end workflow. And now you must have got a general idea of how you can string together tools. These multiple tools that are available in the toolbox, you can string together to form a workflow. So I'm not going to build an entire end-to-end workflow right now. I have built one already for this demo purposes. So as you can see, this is a very minimalistic resequencing workflow or a variant detection workflow that I have here, that I built for this demo purpose. So same thing you have. I assume that I'm always going to get pre-processed, clean, trimmed reads, but you can imagine that you can add all the pre-processing tools to this workflow if you want, but for this workflow's purpose, I'm always expecting trimmed reads. First step is map reads to reference. My second step is indels and structural variant detection from my map reads. Step 3 is local alignment based on indels detected in step 2. So if you have indels and structural variants in your data, that can kind of affect the read mapping, which can then lead to false variants being called downstream. It can affect variant calling downstream. So this is kind of a very key step to do for your resequencing analysis or variant calling workflow if you want, that you detect your indels and then you do a local realignment based on those indels so that you're not feeding data with indels to your variant detection. So next step is variant detection after my local realignment. So there's, again, same as you can see, output of one goes into the input of the other, and then I want my reads track to be outputted from this workflow, so I have outputted this so that I have this file as an output from my workflow. Then after local realignment, I have variant detection. Then I have filter reference variants. That is, I'm going to filter out variant calls which are similar to the reference sequence. So for example, homozygous reference, for example, I don't want those. And then I'm going to annotate my variants with gene information. So I want gene information. I want to know which variant falls under which gene. So I'm going to annotate with overlap information, and the overlap track I'm going to select as my gene annotations. And then I'm going to output a variance track also. So this is a typical, as I said, minimalistic resequencing workflow. And now you're going to see the power of automation. So for example, I have this workflow built. I am going to run this workflow, and this workflow is going to run all these tools for both samples that I have. Now, other concept that I want to introduce right now is batching. So batching is, as I said, workflow. You can run this workflow for one sample, but I want this workflow, this exact workflow, to run right now for 2 samples, but realistically, say for 20 samples. I don't want to invoke this workflow 20 times manually. And this is where batching comes in handy. So batching is when you batch together a set of inputs, then it's going to run that workflow for those sets of inputs or for those sets of samples individually. So I'm going to select this batch because I want to run this exact same workflow for my 2 samples. And when you select batch, it will allow you to select a whole folder of your sequencing reads. So I'm going to run my workflow. I select batch. I select the folder which has sequencing reads. I go next. Here it shows the batch units, and by batch units, it means that the workflow is going to run individually for each of these ones. So at this step, you can, for example, if you don't want the workflow to run for one of the read samples. For example, right now, I don't want the workflow to run for this sample, for example, and you can remove it from this batch unit. So I don't want this. I only want the workflow right now to run for my cucumber line A and my cucumber line B. So I'm going to do next. This is the workflow input. That is the gene annotations. I want to annotate the workflow, or I want to annotate my variants with my gene annotations. I select my reference sequence. And I'm going to save. In my outputs directory is the output from both of these samples. And I'm going to click on finish. And now what's going to happen is it's going to run the workflow. And another good feature is if you keep the workflow open, you can also see which steps in the workflow it completed and which steps in the workflow it's executing. So the dark purple box means the step is done. The lighter box means it's still going. So other thing that I wanted to point out here while the workflow runs is this processes tab. So by default, you have the toolbox tab open, but you also have this processes tab where you see all running processes in the workbench. So running or completed in that session of the workbench. So for example, if you close the workbench and reopen it again, then you won't have this information. But in the open session of the workbench, any process that you have, you can follow the progress in detail in this processes tab next to your toolbox. And right now I can see that both my workflows are done. So another cool feature of the processes tab is right now I don't have a whole lot of samples, but say you have 20 samples, like identifying where the results for each of the 20 samples when can be a little painful, especially if you have a whole lot of data in your workbench. So if you want to just find your results from a particular process, you just click on this arrow icon here and you can click on find results. And if you do that, it's going to directly highlight the results from that particular process in in your navigation area. So this is very useful to find out the results from your data. Now, as you can see, the workflow coupled with batching is a very powerful automation tool, and you do not need to do any kind of scripting or any sort of scripting to achieve this. It's a drag and drop functionality to build a workflow, and then batching is also available once you have the workflow built. And I also wanted to point out that batching is not only available for workflows, but it's also available for individual tools. So if I want to run read mapping for 10 different samples, for example, batching is available in individual tools also. So getting back to my aim for today, what I wanted to do was I wanted to find out the unique variance in my sample A as compared to sample B. So what I have is, if you see here, I have my variance from sample 1 and I have my variance from sample 2. So I have variance from my line A, I have variance from my line B. I want to now find unique variance from my line 1 or line A, and then I want to visualize that in a dynamically linked view in a genomic context. So first, to find out variants that are unique, I go back to my toolbox. Under resequencing analysis, I have compare variant tracks. So I select my variance from line A because I want to compare line A to line B. I want variance in line A which are unique. So I'm going to compare my line A. Comparison track is line B. And I'm going to keep variants that are different, which is going to give me unique variance from my line A, for sample 1. I'm going to save that track. And now if I open this, this is the track of variant, and this tracks has different views. So I want the table view. So I'm going to click on table view. And so basically I have 151 variants, unique variants from my sample A. And this is a table view. And it's going to give you all sorts of information, such as count, coverage, frequency, and everything that you can see here on the left-hand side. But now I want to visualize these variants in a genomic context. I want to visualize them with my sequencing reads, with my annotation tracks. So what I'm going to create is I'm going to create something called as a track list. And to do that, go File, New, Track List. I'm going to select all the tracks that I want in my track list. So I want my reference sequence, I want my gene annotations, I want my sequencing reads from both samples, for example, and I want the unique variants in the ones that I detected in my sample 1. So in this track list, I have my reference sequence, annotations, and sequencing reads, and my variants. And I'm going to say finish. And this is going to give that genomic view that I wanted. So I have my reference sequence, my annotations, my sequencing reads, and my variants in a single view. But now I want to analyze these variants further. I want the table view of the variants to give me more information. So I'm going to right-click on the variance track in the track list, and I'm going to do open this track. And what this is going to create, this is the dynamically linked view that I was talking about. So this is what is it's a very useful thing to have. So, and by what I mean by dynamically linked is now this table view is linked to this genomic view at the top. So for example, if I want now, I can see all the coverage and frequency information, and I'm interested in a particular variant. So say I'm interested in this variant. You click on it, and it's going to navigate you to that particular variant in the genomic view. And now I want to explore this variant at the base level. So I click to go to the base level, zoom to base level, and say if I want to explore this variant, I click on it, and as you can see, it's going to take you to that particular variant in the track, and then you can visualize that particular variant along with all the different tracks and all your different annotations in one single view. So the track list is also another very powerful feature of the workbench. So to recap what we saw how to import reads in a workbench, we saw how to import reference sequences, we saw how to run individual tools, we saw how to build workflows and how to run workflows, and then how to batch samples, and then we saw how to create track list, which is kind of, so the workflows, batching, and track list are kind of the three very good features of the workbench. And my aim for today is done. So I wanted to detect variants from my sample, from my cucumber line A, which were unique, and I did that. I have 151 variants that are unique, and now I will explore these variants further. So some other things to point out in the workbench: the help menu here at the top has multiple tutorials. So if you're just starting to use the workbench and you want to get more familiar with the workbench, you have a bunch of tutorials that you can take complete advantage of, and the tutorials tend to focus on different aspects like de novo assembly, resequencing analysis, RNA-seq analysis, etc. And so tutorials under help, and then please visit us at kaizenbioinformatics.com for product manuals. You also have the tutorials over there, training videos, and PDF format of the manual. Please visit us at kaizenbioinformatics.com, and there you have product-specific pages for each product. And thank you very much for joining me today. And we'll take some questions now.
Hi, thank you very much, AJ, for this very informative presentation. So as AJ just mentioned, it's time for QA. And but before we get started, I just would like to remind you that how to submit questions. You can submit the questions by tapping them in a question box, and which is the bottom right of your control panel. And so we currently have 3 questions that we can start answering, AJ, when ready. Let me open this up right now. So the first question is, how did you produce or obtain the annotation file that you imported right after you imported your reference sequence?
So usually annotation files, as in you can acquire them from public resources, such as NCBI, for example. Sometimes you have specific crop-specific consortiums, for example, for corn and soy, and usually those kinds of files are available in public sources, or sometimes with private companies you have in-house pipelines that build these files. But so long as they're in the standard GFF format, which is the standard, then you can import them into the workbench and make use of them.
Thank you. The second question is, after overlapping of pair-end reads, we have 2 files, overlapped and intersected reads. How can we use intersected reads for further analysis? CRC Bio need 2 separate files for PE.
I'm not completely getting the question, but yeah, what I think you're trying to say is, as in, does it require 2 files to import paired end reads? So for the answer to that is yes, as in if you select paired as your read format, and then it does require 2 files to be selected, because and usually most of the sequencing platforms, when they export or when they output paired end reads from the machine, you usually have the forward and the reverse files, which is usually how it is for most platforms. Sorry if I didn't answer your question correctly, but definitely if I didn't, then you can always write to our support team at bioinformaticssupport@kaizen.com if you want further information.
Great. So we have a third question. how many genomes are possible to compare at the same time, or the maximum?
But if you're talking about genome sequences, complete whole genome sequences, that you cannot do in the workbench right now. But if you are talking about comparing variants from those different genomes, like you want to detect variants and then compare the variants across different genomes, then there is no limit as such. Like you can compare variants across 50 or even 100 genomes. You're only limited by your computing power there. So depending on your system resources, like if you have very small amounts of system resources such as RAM, then that might limit you. But the workbench as such will not limit you for those kinds of comparisons. But yes, genome sequence comparison, if you're talking about that, I think that's not possible right now in the workbench.
Okay, so we have a time just for one more question, and we have a lot of questions, AJ. So I have to select the one, I guess, that will work here. So there is a question about how to compare variance between two groups of samples. Example, 4 samples with the same phenotype in one group.
As in, so as I said, right now, I just, for this demo purpose, I had 2 samples. So I selected a tool called compare samples, but there is a compare samples within groups tool that you can use for the purpose that you're talking about.
Okay.
And
Wonderful.
it's under the resequencing analysis toolbox.
Great. Thank you very much.